Hello! Welcome to Microbiology
Hello.
Online Journal Club. I'm Faz Alam, and this is my colleague and co-host Danny Chan. And we're
Hi.
here. Oh,
We
sorry, no,
talk
you.
about all sorts of microbiology papers, but particularly in this time, we've focused on the COVID-19
Mm,
world
yeah. So,
and trying to bring to bear some of our prior experience in infectious disease microbiology. Um, and yeah, just to
yeah,
get a better understanding for ourselves, I think, right? I'm
yeah,
very motivated to like read these papers for myself, like just so I'm like a little bit more aware of what's going on. And if people ask me questions, I can respond reasonably.
yeah. I mean, we both read papers for a living, basically, and
Hmm.
this was a way for also to include you, the audience, in this process of peer review, and and nowadays open peer review is this entire nebulous process that involves everybody reading papers. So
Yes, yeah.
this is a
Yeah, and we're structuring it in a way that we want people to join in, right? Like,
yeah,
if we have a Twitter account and if you tweet us the papers you want to discuss, then we will discuss them during this choosing phase, and then in the following week we'll open one paper up and we'll dive into the figures one by one.
yeah. Uh, so yeah, we've got. I mean, this has been an interesting week for COVID-19 news. So
Hmm.
I mean, in terms of last week and last time on this we talked about dexamethasone, and in an
Yes.
amazing novelty, they after releasing the press release, not to like they released the actual full paper with all the data, which it's such a
It's really
yeah,
nice.
it's
Yeah.
such an amazing thing because we've got. I mean, we keep hearing some great things about Moderna. Like their press releases look so good, but they don't have the actual full paper yet, so we can't. And oh,
Yeah.
Inovio.
Well,
Sorry.
I mean, I wonder if it makes a big. I wonder the dexamethasone study that was funded by the NHS,
Yes.
right?
Yeah.
So I wonder if that's also helpful because it's not just like an academic lab. There's like a larger organization sort of working behind the scenes to make it.
Well, I mean, I think also it's a big thing to with big pharma. So I don't know
Mm,
if you heard that Inovio had some real like issues in the news. So we talked about the Inovio vaccine, the one that electrifies you while giving the Chadoc's DNA. Um,
yes.
but they had like some real arguments and court. They were trying to sue some people about their studies.
Yeah.
Because apparently like the people. So I need to look at the data, but it was pretty funny. But because again, I really
Hmm.
like the idea of the vaccine, or I think that it's probably like it's
Right.
electrocuting yourself to get vaccinated is something that I think is not going to go down very well with the general public. So
Oh, it's very futuristic. People like some electricity. That's like the the ion air or something like
yeah,
that. There's power in
it's
electricity.
both very futuristic and very 1950s. Very
But yeah, but I mean, companies they have a bit of a different incentive when they're publishing, right? Um, like they just have to expose their data to the regulatory bodies to get past that hoop, right? And usually the publishing, I think, is driven by, well, yes, they want to show that they can engage in the academic literature, but also if they're using investigators who have academic careers, then those people need to publish, right? And like that's definitely the push. Yeah, because I know from like my experience of like reading clinical study reports, that's like the actual trial that's like behind the scenes, right? The confidential document that the pharma agencies have. They embargo that data, right,
mm,
for a really long time before they release it into publication. And even then, right, like uh, investigators who are publishing, they have to sort of negotiate, right? Like, can we show this figure? Can we show this
yeah.
figure like this? Right? There's lots of that. So a study like the NIH, like they don't have to worry about that sort of bureaucracy. I mean, maybe a different type of bureaucracy,
But
but like
yeah,
not the bureaucracy of secrecy.
yeah, they don't have to worry about like annoying their shareholders and like because
Right.
they
Exactly.
the
Yeah. Tipping
NHS
off the market or
yeah
something like that.
they're allowed to publish
Epidemians.
bad data basically. Whereas with farm you really you have to everything has to be like top of the line if I understand it. Where
Yeah.
you got like
Yeah. Well, we saw that with remdesivir,
mm,
right? Like when the original study that got published from China, right? I feel like it came out and immediately there was a press release that came afterwards saying, "Oh, but there's another study and it's larger, better powered, and it has promising results," right? So like that timing was like done in that way. So yeah, to minimize the impact
yeah
to their shares.
Oh yeah, I mean the remdesivir news of like America buying up all the stocks of remdesivir. I
Mm,
mean, this is interesting to me because remdesivir. I get the impression there isn't really much remdesivir in the world because there's
for
so
sure.
the trial that discovered dexamethasone recovery trial is trialing multiple drugs, and one of the drugs was
Hmm.
supposed to be remdesivir, except Gilead just didn't have enough. So
Mm.
it makes me wonder. I mean, again, now we're getting to that point where we're thinking about the entire production process of like
Yeah, for sure.
how are we going to produce these drugs? How are we going to produce these vaccines? I mean, that Inovio lawsuit was they're suing that one of the people who they were going to get to manufacture the vaccines because there were like delays on it. So
Ah.
and that caused
Not
like
they weren't making it fast enough, like according to a contract or something like this.
yeah, which I mean
Hmm.
that in contrast you are basically promising to do something. So if you like kind of don't do it, then it's kind of understandable when someone goes, "Hang on, here's my, I'll get my lawyer on you." But
Right.
that sort of brought on shareholder lawsuits because shares were like brought down by this news. So yeah,
Man.
it's tough to like. I mean, from one of the things I'm learning about this coronavirus is investing in pharmaceuticals is a really tough game and is
Oh
really
yeah.
fraught.
I mean, that was, we know this from grad school, right? Like no one invests in new antibiotics anymore. There are
Yeah,
classes of drugs that just aren't popular because of how difficult they are to make.
yeah,
Um, and I mean that has real effects for like the health system, the healthcare system as a whole, right?
yeah.
So this NIHS, the dexamethasone study, this is the effective dexamethasone in hospitalized patients with COVID-19 preliminary report,
Yes, it is.
the recovery group.
So yeah, that's uh,
Yeah, so accepted for publication, still like sort of drafty as a paper.
yeah. So it hasn't been like fully peer reviewed yet, but
Yeah,
it, most of the
but the manuscript is together.
yeah, manuscript is together. They've got like some supplementary material, and of course, the original trial document for the recovery trial has been published. So we've
Nice.
got
Oh, that was published with the press release, right?
yeah. I think it was published a bit when the trial started. So they
Oh yeah.
basically put out their entire plan, which is kind of a nice practice to do because that
Sure. Yeah. And now you're saying too, I remember we were talking about this last time, like now that they had the manuscript together, we can also see if there's differences, right, between right the
yeah.
pre-published methods and anything that they ended up doing.
Yeah, because I mean, when I was working at like publishers, one of the things that we'd screen, one of the main forms of skullduggery you'd find if people are messing around with big crinkled trials is seeing like how how the outcomes change depending on like,
Mm,
and so sometimes there are very reasonable reasons for changing the outcomes. Sometimes it's sometimes the experience change. Sometimes they need to find they've just something didn't occur to them. But sometimes you see things where they just like spam all these outcomes and then pick one that it looks good and the rest of them don't, and then you never
yep.
hear about the rest of them. And so there's this thing called researcher degrees of freedom where you can actually
Mm,
just through like sheer like probability, if you do enough tests, one of them will become uh, significant, and if you can choose that one and show that one and not the rest of them, you can make something that looks significant that isn't.
huh. I've actually never heard it described that way as researchers' degree of freedom. I definitely heard cherry
Yep,
picking statistically significant results.
yep,
um, and it feeds into that, not to go down too much of a tangent, but it feeds into that whole reproducibility issues, right? Because if people are doing this and not reporting everything, it becomes very difficult to interpret what's going on, right?
yeah,
What's being interpreted. There's an inherent bias, right, to report these positive results, and of course, in statistics, there's a probability that some proportion of those will be incorrect.
yeah. Uh,
Yeah.
and like it's trying to look at. I mean, my favorite one is like this one drug where the outcomes were basically like either normal values of pneumonia or you ask the patients whether they think they had pneumonia, and
Mm,
they went with the one
yes.
that gave them most significance. Was this okay? So does this patient have pneumonia? Do you have a pneumonia? Uh, yeah, yep. Or he
Yeah.
goes, no, not really. Or I don't know. That counts. Which is
Yeah, so this dexamethasone paper, it has like a few tables, and then some survival curves for people, or not like survival, but sorry, the
Kaplan-Meier distributions. So
Kaplan-Meier,
those
yeah,
the
Kaplan-Meier
weird
curves.
boxy like line graphs. So
Mhm.
let's see if I can pull that up on the screen. So I've got a screen share now, because I'm trying to be fancier this time. Uh,
Ooh, nice.
yeah, trying to be fancy, but Yeah, so I mean, actually, that because the thing about Kaplan-Meiers is they look more like normal graphs the more people you have, and this was such a big study that you can see like that it
Oh
is
yeah.
starting to look like a real line. Um,
Mm-hmm.
and yeah,
Mm-hmm.
they have just so many people in this study. I think this is the biggest study with like something done like this ever on like one single disease. Because usually like with like a study on anti-inflammatory, you'd have to test on all these different bacteria, all these different viruses that have slightly different
Mm-hmm.
etiologies. So you don't necessarily, so there's going to be a lot more variability in your results. So having like something like this could be very interesting to see. Like, I mean,
Sure.
especially in terms of like whether it's going to be applicable to other like bacteria and viruses, because then that's a really big can of worms.
Hmm. I mean, there's still found, right? This is still, like they could have. I guess we can see that in the study design. Like we could see to exclude. I'm always
Yes,
interested in seeing exclusion, yeah,
yeah,
criteria.
because sometimes then
But
that's
yeah, we don't have to dig into it now. That's one thing on the docket, right?
yeah.
Let's see. Oh, talking about talking about physics. I was looking at this paper, structural basis. Oh, structure-based design of antiviral drug candidates. Because
Ooh,
I don't know, I was just looking
oh,
for
okay.
looking for
That's
like other things. Did you find it? Do I have to send you a link?
so it's in the Zotero, right? So
Oh yeah,
structural
it's in the
based. Yeah, got
Zotero.
it. Yeah.
Yeah, Dye
So
et al.
yeah, I've got it. So this
So
is
this is interesting because I mean it's very we haven't done a structural, that was one of the ones that we could have chosen, right? The structure of the receptor. But this is kind of showing that once you have that structure, you can try to predict and understand how small molecules will bind to it, and that might show us different therapeutics. So there's the data here is cell line. They don't really go too relevant. Um, but I think it's interesting from the standpoint of like just designing drugs, right? This is the interesting topic.
so hold on. This is the annual review paper, or do I have like a different
Oh
paper?
no, this is the annual review is I have one from Science. It's Dye et al.
Okay.
Um,
Di. I mean, that's a great name to have on a paper for a drug.
DAI, DAI.
DAI. Okay. Uh, that's oh yeah, got it now. All
Yeah.
right. Yeah. So yeah.
It was
Now.
actually I was finding papers through that annual review, right? Because like I was like, oh,
Ooh.
here's a recent review, and like let's see like what's going on in the land of small molecules. Because we haven't, right? We've talked about
Yeah.
vaccines. Remdesivir is a small molecule. We talked about that at the very beginning. But how are people finding other small molecules, right? And this is actually like a huge mode of drug discovery, right? Where
Mm.
people try all the small molecule libraries and see what sticks.
Yeah, you got some nice fancy like pictures, and you got some. Yeah, it looks pretty interesting.
Yeah. Anyways, that's one I'm gonna throw out there.
Okay.
How about you? What's next on your list?
Next on my list is yeah, so Pfizer have a DNA drug. So
Mm.
sorry, an RNA drug. So again, this is quite an innovative idea. They released a press release, and then they also released their a small. It's not like a full study, but a phase half study. I'm not sure that's the thing,
No, no,
but
it's phase 1/2.
okay,
That's
one slash
what it
two.
means. So it's like when they're rushing, they'll merge them together, right? Because like one's supposed to be safety, two's supposed to be like dose escalation for efficacy, and
Okay.
so like they'll kind of merge them. That's what this is.
Yeah, so this is an interesting one because again, it's an mRNA vaccine like the Moderna vaccine that we've heard very
Mm-hmm.
good things about, but haven't seen much data for yet. And
Mm-hmm.
and so Pfizer have just come out with this like big like, well, no, it's not that big, but it's a very interesting like study that goes straight into humans, and
Yeah.
so it's been quite an interesting.
Um, on figure four, they look at neutralizing antibody titers.
Now that
So that's kind of cool.
that's quite cool.
In
That's I mean what you
humans.
want to see.
Yeah.
Uh,
Can't tell if they're using SARS-CoV-2 though in the test. I assume they are.
yeah,
Otherwise, how could they call it neutralizing,
it'd be very frustrating if they didn't. So yeah, you've got like all
Yeah,
sorts of yeah. They've
it's interesting. 4 figures. Yeah, I don't think we've talked about a phase 1 or 2 study yet, right? We've
no, I
talked
don't think
about
so.
yeah, we've talked about a bunch of animal models. Yeah, I don't think we've really looked at human data that deeply. That it
Yeah.
is interesting for that reason. I guess that's for both, right? Both the dexamethasone and this one, human data that we haven't been
Hmm.
looking at.
Yeah. So I mean,
Um, oh,
yeah, go for. I mean,
oh, okay.
this
I
is.
was just gonna plow to the next one.
Oh yeah, yeah. Let's plow into it.
So continuing on my small molecule bent, I found this one, Hoffman et al. Nafamostat mesylate blocks activation of SARS-CoV-2.
Okay.
Another small molecule, and this is different because oh, this is only a single figure paper. There's actually another one. There's something related to this one as well. It's the other Hoffman paper. Maybe we could talk
Okay.
about them together since they only have one. But
So is that?
the in
Yeah.
oh,
Okay.
it's the
Got that one. I got
it's
that
a
one up. Yeah.
cell one. Yeah, both Hoffman. But basically, it's a small molecule that targets our receptor.
Ooh.
um, or like these are different targets, right? Like one of them targets a protease that prepares the ACE2 receptor, and then the other one targets a protease inside the genome of SARS-CoV-2 itself. So these are just like different targets that we haven't been talking about, right? Is this? Oh no, these are both. Sorry, I misspoke. These are both targeting this TMPRSS2.
Okay. T-
So that's in us, I think.
So
It's
yeah.
uh it's a protease in the host that is required to activate the spike.
Okay. Oh right. I see. Ah.
It's kind of like when they were talking about the furin cleavage site. I think it's related. I mean, I didn't dive too deeply into it, but it seemed like the same thing to me.
So yeah.
Um,
Apparently it's already clinically approved, and it's
yes.
so yeah. And it's an interesting idea because I haven't actually heard because usually when they talk about SARS they only talk about ACE2, but the way this
Yep.
describes that TMPRSS2 also apparently helps. So that's an
Yes. Yes.
interesting
I
like
mean, it also opens up this question of, right, there's sometimes redundancy in like a infectious thing, right? If
hmm,
like you're relying on a protease to pre-cleave your spike, there could be many proteins in the human body that do it. But when they did these, they did some it's all cell culture based stuff. But it seems promising in cell culture.
yeah. I mean that's I mean it's interesting mostly to like look at the wet method that people are using to discover these small molecules and to just and like again this really delves into like how the viruses work, which is I guess like kind of what we studied in university. So
Yeah, absolutely.
so yeah,
Yeah,
that
like sort of talking about like the exact steps required to get into the cell and saying then where can you break those exact steps,
yeah.
right? How do you break it?
Yeah, I mean it's like that formula puzzle piece of just trying to like figure out oh which protein is missing or what is what the like each
Right,
steps of it, what's kind of creating
right.
defective viruses and stuff like
Hmm.
that. It's
Hmm.
like
Yeah.
I mean was well for us I think for our generation that's classical microbiology, but now everyone's got fancy genome stuff and like
Yeah.
so
Well, I
I
mean,
mean
like to get to the story, I think you go from big to small, right? It's like there's all
yeah
these like big data collections, you're refining your hypothesis, then hopefully you go down into some sort of mechanism.
yeah so I mean I mean it'd be interesting to do like a double header day of two papers.
Yeah, well,
Uh,
because it's the same mechanism, or
yeah.
right, like it's presumably the same mechanism. I think they're related compounds from what I understood. Like
Hmm.
one just like binds better. So like the antimicrobial agents and chemotherapy one, that's just like they found like a better binder. A
Okay.
small molecule that's a better binder than the one that they found in the cell paper. But I wasn't able to find any animal studies, even though like in the review it said that it was in animals. So like I'm not sure if that's just like personal communication. Like they didn't link, they didn't reference to the animal studies. So I don't
I
know.
mean, sometimes you get that in reviews where they just go like, "Well, I guess so," or sometimes like you get reviews where they don't actually read the paper that they're citing. Uh, which is
Well, I mean, they made it seem like it was like they had a table and it said like where the drugs were in the process, and it said like in vitro and in vivo animal model, and I was like, okay, well, where's this in vivo animal model you speak of?
yeah, that's
Maybe I didn't read the text. Maybe they would have said in the text like
yeah. I
the
mean,
personal communication or something like that.
so, yeah, it might be like that. But yeah, I mean, again, I've been a victim of my papers being cited by people who haven't read the papers or understood them.
At all, yeah.
So I'm
True.
kind of cynical about that, where they go,
They
oh,
could have
this
just been like, biological relevance, cell culture equals animal model.
like LMNL did this, therefore they prove it, and I was like, I definitely didn't do that. I definitely didn't prove it what you
Yeah.
think I've done. It's like I'm like, yeah, I've had that frustration of some like of reading something like, I wish I'd done this. This would have been
Yeah.
great if I achieved this in the lab, but I didn't. I'm not that. Oh, but yeah. Uh, well, what other things? Oh yeah.
What
So
else?
other.
Continue parading them out.
So I've got a library of nucleotide analogs can terminate RNA synthesis. So this again is going to small molecule kind of stuff, but this
Ah.
goes more into the nucleotide analogs part. So it's rather than because the other ones they target multiple different types of proteins, but for like remdesivir and a couple of other like anti these broad spectrum antivirals that were being
Hmm.
developed for multiple diseases, they tend to try to target RNA synthesis. So
Hmm.
so often like these, so there are a
Oh,
couple of things that have been like
a
developed.
lot of mass spec on this paper.
Oh yeah,
I mean, this was like when we did the macaque paper for remdesivir, they
mm,
used mass spec to show that it was binding to the RNA-dependent RNA polymerase, right? And it was being metabolized in the body as well, like how some things get metabolized. I think this is like a common thing for nucleoside analogs. They like come in one form and then yeah, they come in as prodrugs and then they become activated through
yeah.
metabolism.
I think it's because like they get chewed up very easily, and so you have to find a way
Hmm.
to protect them before they go through. And
Right,
also
right.
also I think there is a sort of aspect of targeting towards the viral nuclei specifically, because so
I
having
mean
a
that
pro.
Oh, I'm gonna say like dependent RNA polymerase. That is definitely not a feature of the human body.
Yeah, but I mean, theoretically, I mean, there are like there's some spillover sometimes with the antiviral drugs that causes some like side effects, and one of the
Sure,
because everything uses nucleotides, and so like trying to get one that is like targets specifically and has very little spillover, because when you want to give someone an
sure,
antiviral, you want to give them loads, you want to give them enough to like flood the body.
right.
Um, because again, you need to like not only do you need it to get inside the
Yeah,
body, you need to get inside like cells for it
right.
to work.
Yeah, so
And
like bad nucleoside analogs would be things that also get taken up by like DNA polymerases
yeah,
and or DNA dependent RNA polymerases and yeah, I mean that stuff we definitely have the function for.
yeah, but yeah, there
Interesting.
is a load of mass spec on this paper. So if you like at mass spec, mass
Did they
spec and mass spiel.
It doesn't seem like they do anything like biological.
Yeah, no, this mostly is like playing the numbers by the looks of it. Just
Yeah.
creating a massive big science
And like
um,
merging or like maybe screening through recombinant protein to look for these.
yeah. So I mean,
I'm
part of
not
it's
sure.
like the actual way they test it is the is an interesting part. So trying to figure out. So when you're doing these giant libraries, you want to like have very tight testing tools to try and figure out what's going on.
Hmm.
Um,
Hmm.
but yeah, reading a
Cool.
bit deeper, it's not quite as exciting as the other papers because again, this is even earlier. This isn't
Yeah,
even
this is
really in
even
cell.
earlier. They haven't even done cell culture. It's
Yeah,
like
yeah.
yeah.
This is like a pure like chemistry
But
paper basically.
but it's cool from the technology standpoint,
Yeah,
right? Like if we wanna if we're thinking about like spreading over different technologies and talking about them more, then this is a technology that we could talk about. Okay, I have one well, also Lucy et al.
uh,
ALSOUSSI. A
EL.
potentially neutralizing antibody protects mice against SARS-CoV-2 infection. And so
I
this is using, we talked about that mouse model that was reported a little while back, the one that uses adenovirus to infect the lungs of mice.
Oh wow.
This
So
is
what's the name again?
actually
I completely
oh
spaced when.
the author is Alsouussi, also Lucy. A potentially neutralizing antibody protects mice against SARS-CoV-2 infection.
Right, got it.
JI.
Yeah.
Um, yeah, so how we talked about a few weeks ago, there's a mouse model now for this. Well, we actually talked about two mouse models, right? One where they
Yeah.
adapt the SARS for the mouse, and then, but this is they use the one with the adenovirus to infect the mouse lungs to give them ACE2. And then they think they screen through an infected mouse for monoclonal antibodies, and then they purify it back out, and then uh, passive transfer them into the mice, and they show it has protection.
Okay. Oh well. That's interesting. I mean, that's interesting way of generating these antibodies. But it's a great opportunity to first talk about a model that I think is kind of bonkers, but kind of
Yeah.
great. Um,
I mean, it's the only one we have. Like, it's the only mouse model, one of the two mouse models out there.
yeah. Although,
Yeah.
I mean, technically we could actually do all this with a hamster model, but because
Right.
hamster models have like an ACE2 gene as well. But again, this one it has got the human ACE2 specifically. So theoretically,
Yes.
this way of screening could create antibodies that could be used in humans.
Yeah. But it is also interesting to think that they are making mouse monoclonals, so like that's like unique for mouse immune responses
Yeah.
potentially. Yeah.
So
So of course the mouse monoclonal that comes out of a mouse is going to protect mice. Sort of, right? Like
yeah.
there's something kind of very obvious about that finding that isn't totally translatable, but promising, I think. Promising.
Yeah, because I know we've wanted to talk about passive transfer and like with convalescent serum and stuff
Yeah,
for a while,
and there
but
have been, right? I think there have been studies that we looked at previous or like in the selection phase that was
yeah,
looking at monoclonals isolated from humans and then doing them in cell culture and showing neutralizing ability. So this is like it's a different setting, right? The mouse, but I
yeah,
think that again, like interesting information. I wonder whether or not we'll have like because this is also one of the in the running, right, for COVID-19 therapeutic monoclonal antibodies,
yeah.
and we
I
haven't really
think
talked about that at all.
yeah, I think like the yeah, it's convalescent serum is one of those things that I think is kind of an interesting one because again, it's one of those treatments that can be used in the late stage. That you give
Hmm.
like people in the late stage like lots of intravenous antibodies and then hopefully that can help like fight off the infection. But then
Yeah,
again,
yeah,
it's used in that phase because it's very hard to give it like kind of casually. It's
right.
but it's
Well, I mean, that's from the convalescent serum point of view, right? Because that we're talking about like someone else's serum, but like
mm,
there's also the idea of just making the monoclonal,
yeah
right? So
and
like that's like I actually picked up that one as well, tocilizumab.
yeah
To-
tocilizumab,
tos-
which is for
Tocilizumab.
IL-6.
Yeah, I mean, so like a different mechanism of action, but it's in the class, right, of biologics, right?
Yeah,
An antibody that binds to something. But these monoclonals, presumably, they're binding to the virus itself, some motif on the virus. I think they even show it binds the receptor binding domain.
yeah. I think a lot of these papers are focusing on the RBD as the
Yeah,
slang as the kids are calling it.
as the kids are calling it,
Kids are calling yeah. Uh,
the mature scientific children.
yeah, I mean, all the coronavirus memes are hot. I
But we
mean,
have we haven't talked about like monoclonals and the process of making them. You know, that's a huge part of the pharmaceutical industry, is monoclonal production.
yeah, um, yeah. I guess I mean, yeah. So to Silizumab, let me look at that paper again because we have talked about it before, but I thought that was quite an interesting paper, especially in light of the remdesivir news, because I think that
Yeah.
was in
So in the in the Zotero, I also put up a tocilizumab one. Um, it's it's a retrospective, I think, study.
so
It's
for cytokine release syndrome in hospitalized COVID-19
yes,
papers. So that's
yes,
quite interesting. Uh,
correct.
so yeah,
So I think they're looking back on a cohort and seeing the effect that it had. I
yeah,
think.
I mean, there's a lot because we haven't really done much observational studies. because there's a lot of like things that can happen. And I think this is they've got like a review copy because I can see like notes. Let me check this because then I
Oh,
because
this Q1, Q2 stuff in the corn.
yeah, it. I mean,
Yeah, I
yeah, it
mean,
looks like these look like basically. So in the corner you can see these red letters, which
hmm
usually when I see those, they indicate that the review has something to say. And
I think they got flattened though onto the
yeah,
thing. They're
so
not
it could
actually.
just be like a production issue where they forgot to take them out.
Yeah, I think a production issue.
But it's a good
Uh,
thing that they're there so that I know exactly which things to read and go, maybe I should look into this a bit more because
yeah,
maybe
yeah,
someone else
exactly.
has got something to say about this. Like, I mean, it probably there's author notes because there's a lot of them around the funding notes and the correspondence. So
Mhm.
it is then
But I
like the
mean, this also harkens back to a couple weeks ago. You wanted to maybe do an immunology focused, right? Where they looked at
yes,
some omics style version of what cytokines are involved in this.
yeah,
Yeah.
yeah. So I think that touches on that pretty in a pretty interesting way. And of course, there's effective treatment of severe COVID-19 papers with tocilizumab as well, which is in the Zotero.
Yes. I think
So
we this was from last time we had put this on the docket. And this is like a
yeah,
this is also a retrospective, I think.
yeah. So that yeah, I think they're both retrospective. So I guess pick your punishment with this one. So um, yeah, I mean, again, the immune response is quite an interesting part of the the whole COVID-19 story. Um,
hmm hmm And we haven't
and
talked too much about that one, that
yeah,
part of it.
we so
I
I
mean,
mean,
there's so many aspects we haven't talked about.
there
Yeah.
really are because it is just massive. There are so many papers out there on COVID. Something which I believe that my next pick is going to discuss a bit more. So my next pick, it's a bit out there, but it's basically so methodological rigor in COVID-19 clinical research, a systematic review and case control analysis.
Nice.
So this is a bit different from usual papers because this is a
Yeah.
paper where the researchers were reading papers and then
Meta science, I feel like people call this sometimes.
yeah, meta science. So essentially, what they did was they read a bunch. They were trying to read a loads of COVID papers and then scoring them for whether they mention like say, whether they mention like basic things like whether they did a sample size calculation or whether they included data on like their like sorry, whether it's a retrospective study or whether what types of articles. So like uh, it's quite interesting to look at from our perspective because we had to trawl through these papers and saying looking at like how 86 so of the articles 86% are like retrospective studies or
Yeah.
and like looking at the methods of like SARS-CoV-2 analysis and like they're like five which only look at physical exams which are basically like does he look like he has SARS-CoV-2 you look like you have SARS-CoV-2 and
hmm
so is so I
But
mean
it's like a
it's
nice overview of like how much there is out there, but also
yeah.
what might the use be of having so much, right? Like some of it, there's lots out there, but it's not all useful stuff.
Yeah, I mean, like this is almost like the first step you do in a meta-analysis, where you look at the quality of the data and pick the ones that you want to use in your
Yeah,
meta-analysis.
yeah, yeah. This is like the pre-meta-analysis. Like before they go and synthesize the things, they're just like, yeah, picking the ones that are going to be useful for them,
Yeah, I'm
choosing good
imagine.
criteria.
Yeah, I'm imagining these poor researchers reading through these papers, going, "I don't believe this. I don't think anyone else will believe what I have to deal with to get any." And it's like published, and then like their supervisor, yeah, okay, sure. And now, because yeah, again, looking most of the papers in COVID-19 seem to be like well, like retrospective or just like very much not the same kind of quality that you would expect from a published paper.
hmm
I mean, I work with a publisher, and we tend to get we've got a lot of like papers where COVID-19 is tenuously linked to something else, whereas like so we created a computer vision like kind of camera system for looking at pneumonia. Let's
Right.
put COVID-19 in the title because that's got something to do with pneumonia. And then the only mentions of COVID-19 you'll ever see are in the introduction and then the discussion, and nothing in the methods or anything else. So,
Yeah.
um,
It's
so that's
interesting. They see like oh, they even look at time to acceptance.
yeah. So apparently, yeah, time to acceptance. COVID-19 articles are getting accepted very, very quickly. I mean, much
Yeah.
quicker. So 13 days is it's yeah. They either have like really super powered reviewers or really terrible reviewers because either
Hmm.
you get someone who reads it immediately and goes, or someone who goes,
Yeah.
eh, eh,
That's part of the suggestion they're making, eh? It's just like maybe things are happening too quickly.
yeah. I mean, it's maybe
But they
like
also say quality, and we can talk about the quality of papers.
yeah. I feel like again, quality is it's a very like hand wavy kind of term because again, it depends on who's doing the analysis, their opinions are.
Hmm.
So, uh, I mean, what believe is a very quality, but then again, like they do look at oh, they look at impact factor as a measure of quality, which
Oh, that's so tough. I
no,
feel like they're trying to phase that out from reviews, right, for jobs and stuff. It's not good to
yeah.
replicate that
So
structure here.
yeah, I know they've actually got this declaration called like the Declaration of Research Assessment, DORA, which people sign
Hmm.
on, and DORA basically says, hey, if you're applying for a job, we'll actually read the papers you've published before before making a judgment on you.
Yeah,
It's
instead of
like,
just being like, sorry, not top tier journal. You're not accepting. Yeah.
sorry, your numbers are lower than what we want. And like,
Yeah.
oh, this person's numbers are really high, but they're a historian, but their numbers are really high. They're not.
Yeah, I find that very troubling actually that they use median impact factor here. Well, I mean, it's
Yeah,
just one of the metrics, but it's a bad taste in my mouth, I think.
yeah, yeah.
Just
Again,
especially from the in the domain of like sort of meta research, I feel like that it's not, it's only a correlate of something, right? And you don't know what factors correlate necessarily with that impact factor, right?
well,
There's
yeah.
confounding variables that aren't being assessed.
And the thing is, they
Um,
can only get the impact factor from the journals. It doesn't actually, they can't get any of that data from the article itself because in order to do that, they have to wait 2 years, no, 3 years to get the full like kind of data of how
uh,
many who's citing. So they're basically looking at all the
right.
places that it's been published and how often that's been the impact factor of that. So this tells you more about where these articles are being published, and so whether
Yeah.
high impact factor journals are actually just giving things a free pass, going like, okay, this is very newsworthy, so let's just like give things a free pass. This is,
Yeah.
and you can sort of see that reflected in people's submissions because they're thinking, well, this could be my first and only Nature paper, so we'll just like try and find some way to crank COVID-19 in the title, and then we can maybe get this through. And once you have a Nature paper, suddenly your career is made. So
Yeah. It
yeah,
makes a difference to people.
it it really it shouldn't. The quality of the paper should make a difference. And yeah, Nature
Yeah.
has a quite good review system, and they usually can get high quality papers, but you should read the paper before you decide. That's the
Yep.
whole idea behind this podcast.
Right.
Podcast,
Right.
YouTube channel, like uh, it's
Yeah.
like
Yeah, I mean,
yeah,
I don't I definitely I think it's great that we brought this paper out. I don't know if I want to discuss it like
no,
for a full hour, but just like the points that we've made already, looking
yeah,
at it, I just feel like okay, I don't know, especially because it's not actually extracting, it doesn't follow through with the meta-analysis,
yeah,
right? It would have been nice if they, even in just one of these categories, case series, cohort, case control, or diagnostic studies, if they opened it up and they then told us something about, right, the data that was inside of all of the papers that they collected.
yeah,
Um, they just wanted to judge the papers,
yeah.
but then they don't even extract out the information for us, really.
Yeah, and I like judging them for judging this paper, so that makes me therefore more
Oh yeah,
superior.
absolutely. Judge the judges.
And whoever judges us, you win.
So yeah, in the comments below, feel
Yeah, just
superior.
yeah, just say like, yep, I disagree, I'm superior, and you win the internet.
Okay, so I had said that there was a paper that I was looking at a different therapeutic. I thought I picked it. I hope maybe I didn't. I thought I picked one that looked at small molecules for a protease, but I'm not seeing it. So maybe I didn't.
So I mean, we already talked about the TMSS.
Yeah, but that's like for a protease on the host, but there's also a protease in coronavirus, and
Oh,
in the review they had went down the path. I guess I didn't put it in here. Okay, but I did put these ones. Lee et al. Role of air distribution in SARS transmission. So this is from classic SARS. It
ooh,
was published in 2005, but I guess it's sort of paired with the Park et al. Coronavirus disease outbreak in a call center in South Korea.
okay.
Um, it was early. It was published early, like eight. What is that? Oh, maybe that's
So yeah,
not.
published in 2004 by the looks of it.
Yeah, yeah. So one about SARS in 2004, and that's in a hospital.
Mhm.
And then in emerging infectious diseases, there's like a similar type of study. Like these are like air studies, right? They're looking at like the airflow inside of buildings and trying to say like because the airflow was like this and we saw these people get infected, we have reason to believe that it flows through the air. And I was just bringing this up earlier that it seems like there's the safety of being outside, relative safety of being outside versus
Yeah,
being indoors. Um,
yeah. I mean, that's interesting because again, like the guidance we're getting for COVID-19 is going outside, but like keeping your distance is
yep.
better than even if you keep this going indoors because I guess you get more recirculated air. There's less air movement, so
Hmm.
like the path between like so if there's wind, then the coronavirus will be like diluted. It might be blown around a lot. But if indoors we don't have any wind, the air's quite still, so kind of it's more likely to find its target if I'm talking to you directly. So if
Totally.
you
Yeah, totally.
so I mean that's kind of physics, but it's
Yeah.
one
So
of
again, maybe these could go together, right? Both the one from classic SARS and then Park et al. Corona disease outbreak in call center. They're both the same. They have like a floor plan map.
yeah, oh yeah, I love it when they have that. So yeah,
Yeah, of like who got it, where they were sitting.
oh
Um,
gosh, yeah, this is interesting. It's one of those things that is would be it's one of the things that would be very hard to get through today. Well, it's just because like of like kind of the identifiable information. Because now in this era with the internet, uh, people get very paranoid about how much because the amount of information you need to identify someone is becoming much lower.
oh, sure, yeah.
So
You infer from these seating plans and timing of disease, like who had COVID in the office.
I
I
mean,
mean,
yeah,
people who work in this office will know just by looking
oh
at
yeah,
this, just by looking at this map.
yeah.
I can't
And,
believe they gave it to me.
and that's a problem if they see oh we've identified patient zero. Oh great, I'm going calling up my lawyer. Excuse me, I need to sue this person
Right.
for giving me COVID. So yeah, that's kind of interesting because it is like one of those because it could potentially be one of those historical artifacts that we never see again because of the way that we have to handle private information in the tw-
Mm,
2020s.
maybe. Yeah, the people don't release this anymore after a while.
I mean, you'd think so. I mean, if I was at a publisher and I saw this, I'd be like, hang on, wait, this
Mm,
could lead us to be somewhat vulnerable because,
for
or
sure.
it could give away someone's information and really screw up their careers and lives. So trying to be
Yeah.
balance that, and we'd
Hmm.
probably have like a network diagram instead, or something else to like kind of uh,
Yeah.
sanitize
Yeah, that's right. Yeah.
information.
Yeah, because you could still understand if you even just represented like distance from these people, right? You didn't have to actually show like that they were the third row,
Yeah,
three tables down, like that person. Yeah.
yeah, because I mean,
Yeah, it's
yeah,
interesting to think about it.
it's one of those ethical issues that comes up when you're like working a pub like will this mess up someone's life is one of the things that you should always think about when publishing a paper. It's like
Yeah.
and the answer is if the answer is yes, maybe you should rethink it. You
Hmm.
should, but yeah,
Totally. I mean, again, like we've said this before, right? There's so many different ways to evaluate a paper, right? This is another axis that people might be not be thinking about, but there's an ethical dimension to publishing certain types of data.
yeah, and it causes so much problem because there's so much that level of shielding means that articles like the Surgisphere papers where they pretty
Mm.
much like nobody knows where their data comes from, and they could get away with that because again we're getting used to that because part of the way our system's working is to make sure that we don't see people's individual information and try to protect it, so we don't necessarily
Right.
see where the data is coming from, which means that someone could step in and abuse that system, which is
Yes. A little bit of
uh,
it. There's a trade-off. Yeah. But again, it's like this is like bad actors in this sense, or
yeah,
maybe sloppy actors as well.
yeah,
Bad and sloppy. Yeah, I mean, maybe this is definitely very different data than we've seen. Like
yeah.
if you look at the classic SARS one, they have these like airflow diagrams, air temperature distribution in two vertical planes.
That's actually
um,
really, I mean, yeah, that's really cool. I mean, I remember like when I was like doing my PhD a long, long time ago. So people were doing like GIS, like where they're in hospitals mapping like areas and
oh
trying to
yeah,
look at airflows and stuff. Uh, so, uh, so they're actually employing a professional like geography major to do that because they
sure,
knew how to
sure.
do all the kind of, because
Yeah.
yeah,
And
it's really
I pulled these papers because right like because of this idea like so much. I mean in New York City like they're reopening all these different types of businesses, but they seem like they're avoiding all the indoor stuff, and I wanted to see like what data was out there to talk about this sort of thing. And really what you get is when you're looking for answers to this, lots of case studies, like infection
yeah.
case studies. Yeah, like this is one of them, but there's a few other ones out there as well.
I mean, I've seen some crazy infection case studies. My favorite ones are when like this one guy got infected with the fleshing disease in his bum, and this I gave away the twist ending to the paper, unfortunately, but the paper was
Yeah.
just them investigating until they found it out. And
Oh, like where did it come from?
where did it come from? And they discovered that like this one person like who carried the disease only in that area somehow spread it
Yeah.
through the air, which is
Mm, crazy.
somehow how would like the only disease that's been documented to spread through farting. It's not. There's an even worse one where this poor nurse was infected the same way, and they made her do jumping jacks and had petri dishes that set at specific distances
Oh
to
my
measure the
God.
range of the like
Interesting.
how far they could get the bacteria to so they could estimate like how the essentially it's estimating social distancing. That was
Yeah.
like one of those
Yeah.
papers where they. So I mean they
Well,
used to have
I
one.
mean that's part of this data too, right? That I find interesting is
Yeah,
like, yeah, it's sort of supporting how far you could get. I mean, there's like the controlled experiment style where you, as you say, get someone to do the action that you think is spreading it, and then measure how far it gets. But then
yeah.
you could also do the observational style, like see in a space who got it and like what their common characteristics might have been,
Yeah, I mean, in the
geographically speaking.
1950s, there were like a bunch of these papers that were set in military bases where they'd have all because again, military recruits they get loads of disease. They don't get, but they have a very active life and they are often in close contact. So they basically get the equivalent of fresh as flu every time they get back, and
Oh my
so they
goodness.
get and since they know where everyone is like kind of sleeping, they actually mapped out the distances between bunk beds, and there's this intervention where they move the bunk beds slightly further apart, and they reduce the amount of outbreaks. Uh,
Nice. I mean,
so
that's such a good stat. That's like so relevant, right? Just like in the general concept to where we are right now, right? Like
yeah,
social distancing and stuff like that.
I mean, yeah, I guess papers go through cycles as well. So like
Yeah.
where the generation that read those 1950s papers have now moved on, and then some of the knowledge gets lost. Uh,
Right.
which
Oh,
again,
and I just realized when I was saying social distancing, I heard that we should be saying physical distancing,
physical
right?
distancing.
Yeah, because it's like more accurate, right? Like there's nothing
Yeah,
really like social about standing further away from people. It's just like it's like a physical metric.
yeah, of course, physical distancing is much more accurate because social distancing is like I've unfriended him. Ah, no,
Yeah,
we're
exactly.
socially distant now. We're not talking. We're sat very close, but we're not talking. uh, yes,
Okay, was there
physical
anything
distance.
else from your list that you wanted to bring up?
There wasn't really anything else from my list.
I'm trying to see if there's anything else in my list.
I mean,
There's that other,
I
and
mean,
then nothing that I found in this round. But then again, there's the stuff from the past, right? We could talk about the actual creation of the mouse model. Um, We could talk about any of the human. We can talk about the cryo-EM structure of the spike. I think
Yeah,
that's still good. We can talk about the immune response. We briefly talked about, right? We said that. Um,
I mean, I feel like the
the immune response.
dexamethasone paper and what other ones were, because we talked about some really good papers this time, but like in terms of the
You want to do human
yeah, do
study?
humans like on go into humans now. Uh,
Yeah, because again, we haven't talked about a phase one or two. So that's the oh, there's like tocilizumab, right? That's
yeah,
one. There's the dexamethasone, and
yep,
there's the mRNA vaccine. Those are
yeah,
three of them that are in humans.
yeah. I think those three studies actually cover the variations of things that we wanted to talk about. So I mean, apart from
Hmm.
the small molecules, but I feel like we can like wait
Yeah.
up on those a little bit. But
Hey, there's always going to be another time. Let's
yeah,
look at this RNA vaccine. I want to see the type of data.
they've got like a convalescent serum panel. They've got
Hmm.
they've I mean they set out their endpoints. They've
Yeah, so
tried the
that one has convalescent serum. Dexamethasone has the survival curves, the Kaplan-Meier
yep.
curves, and then the tocilizumab has some chest X-rays.
Oh yeah, classic chest X-rays.
Oh, well, one of them is chest X-rays, but this one has like violin plots.
Oh, the violin. Okay, right. Let's see those plots. See
The
the
one from chest. Uh,
yes. So yeah, yeah. No, I can. Oh yeah, those look
oh,
crazy. I've never seen those
because
before.
because this one's observational, right? So
Yeah.
it's yeah, so it's less. I actually would rather not talk about an observational study.
No,
Um,
I agree with that. I feel like there's only so much rage you can contain in in one pot. I mean, yeah.
yeah,
Oh,
yeah, because the limitations are sort of I mean, we can say it right now, right? The limitations are like pretty well known in the sense of you didn't control for a whole bunch of things,
yeah.
right? Like you sort of have to just explain the quality of the data set, and it comes with all the caveats of just like picking people. Um,
Yeah, half of the paper will be discussing the data, the other half will be like, but there's we have to take all this other stuff into account with observational. So I agree
right.
that yeah,
Right.
it's not as interesting.
From the mechanistic point, I think the other thing is with these three papers, we won't be talking that much about the mechanism of pathogenesis as much. I mean, maybe we
Mm,
can try to talk about it a little bit, like as a backdrop to like why why blocking certain things might have an effect, but there's no actual data to talk about mechanism in these,
yeah,
right? This is
true.
about efficacy, efficacy
Yeah,
of a therapeutic.
yeah. Um, so I mean, again, the clinical trials give us like the chance to talk about how to read clinical trial studies and
Hmm.
how to. But yeah, the thing is, clinical trials
The
aren't.
phase one, two.
So the phase one two it seems like an interesting one because that covers two of the things that we want to. So we can instead of talking about a phase one and phase two, we can like compress that and be more efficient and talk about the phase one slash two, which
Yeah.
I
I
thought
mean,
was
we
a
can
half.
also talk about phase studies. So actually, the recovery collaborative, that's probably like this would be considered a phase three, right?
Yes.
Because it's a large cohort. They're looking at an efficacy endpoint. Um, yeah, this would be a phase three, I would say. So we
Hmm.
have phase one, phase two. I wonder if we could do both. Do you think that's too much?
I think I feel like we should be
There,
ambitious. We can
there's
try and do
only
both.
two. Yeah, there's only two figures actually in the dexamethasone
I
study.
think we can actually do both, and I think that'd be quite. I'm pretty sure the YouTube algorithm would like that um,
I
because they're
mean,
two.
yeah, it's
Yeah,
like, and it's clinical studies, right? So it's like we can try to talk a bit about like one, two, like those three clinical study stages, and how this kind of fits in.
yeah. I think
Yeah.
that's a good idea. I mean, we might go over time, but if you're okay with that, then I'm okay with that. Because
Yeah, yeah.
yeah,
Four. Yeah, there's four figures here and two figures in the other. I mean, we've done six-figure paper. We've done four-figure papers.
yeah, we've covered like a lot of papers, and
And it's like not like we're gonna spend like a whole bunch of time on the tables of
of like
baseline characteristics or the study design as much.
yeah. So
Yeah, let's do it.
yeah, okay, well we'll do two. It's gonna be a double feature.
A double feature.
I think
Move
that
these into the folder. That's awesome.
yeah, I think that'd be a good thing. I'm gonna write that down in my my book for
Yep. So we're going to be doing phase 1/2 study to describe the safety and immunogenicity of COVID-19 RNA vaccine. So this is a Pfizer study, right?
yep.
And then the NHS study, effective dexamethasone in hospitalized patients with COVID-19 preliminary report.
Yes. So, yeah, I think that those are two very important papers on treatment that will affect people, and I think it'd be
Yeah.
quite interesting. And I
And
think,
and to set the stage, some background about yeah, clinical trials and how those are done.
yeah, I think that that'd be a good. This is something that I think. I mean, I'm very interested in both of these papers because again, this is like, are we going to get out of this outbreak? What are we getting? And they also like again have two like different outcomes they're looking at potentially. Where like dexamethasone, you're basically looking at reducing mortality, whereas the like other paper, you're more likely try stopping transmission. So it's
Right.
so I think the
Well, I mean, it'll be interesting. We can talk about that as well, right? Because the like phase 2 studies are supposed to set up to find endpoints that they're gonna then look at it in phase 3. So we can see like, yeah, we can talk about what they chose to do. Yeah, what we think their phase 3 endpoint might be just from looking at the phase 1/2 study.
yeah. I yeah, I think this is gonna be this is gonna be more than we've done before, but I think that this is a good thing to try out.
Yeah, yeah, and it's also different. I mean, we've been talking a lot about disease models.
Yeah, and I think that we've got like a couple of papers in our like list that we could do as half papers, like just like the
Yeah,
Hoffman papers you mentioned, and that'll
exactly. Yeah, yeah,
help us go through our backlog a little bit, or help us like I won't say back because it's always going to be a massive backlog. It's
yeah,
never going to disappear. It's always going to be increasing, but it'll help us cover a lot more.
yeah.
Um, which I
Agreed.
think will be quite an interesting challenge.
Agreed.
Um, but yeah, I think that's so. I think that we've covered pretty much everything. We've now chosen our two papers. If you at home want to talk to us about those papers, I'm going to put the details down in the description, and
Mm-hmm.
you can contact us on at Defective Brain or at ID underscore ETA,
Mm-hmm.
and
Or the
give us
Micro Twitter Journal Club.
yeah, Micro Twitter Journal Club. We've got the hashtag in the corner if you want to take a look at that
Mm-hmm.
and contribute contribute to the discussion next week. So hopefully
Yeah,
I'll see you then.
sounds good.
Yeah. All right. So bye everybody.